We have covered modes of excretion. We've done structure of the human excretory system, external as well as internal. And in the previous session specifically, we did what is the physiology. The physiology has 3 steps: ultrafiltration, selective reabsorption, and tubular secretion. Right? Now, today in this lecture, we would like to do 2 points. One, how is urine concentrated? How does urine get concentrated? That is the aim that we want to achieve. Okay, na? And third, what is the regulatory mechanism for this process? Right? What are the 3 regulatory mechanisms? So, these 2 points that we would like to talk about. The concentration of urine is achieved by a mechanism by the name of counter-current mechanism. What is the mechanism called as? Counter-current mechanism. So we will see how this is done. Now, first of all, just one small diagram for our recollection of all the events we have studied in the last session, please. So we have Bowman's capsule, PCD. We have this descending limb, ascending limb, DCD, and collecting duct. Yes. So we have afferent-efferent glomerular formation, creation of pressure. If you remember, we studied 3 pressures last time. Right? At the end, the effective pressure was measured at 10 millimeters of mercury, which is causing the filtration to occur. Large volumes are getting filtered here. So glucose, vitamins, amino acids, water, right now, or sodium, chlorine, everything is reabsorbed. Whereas we have urea, ammonia, all these being dumped into the filtrate. Here it goes down. The descending limb is permeable only to water. So from 300 it starts, goes right up to 1200 milliosmoles per liter. Again, the upward or the ascending limb is impermeable to water now. This is permeable only to salt. And then what occurs here is conditional under the influence of ADH and aldosterone. And again the action of ADH continues in the collecting duct also. So we have 75, 5, 15, 4. So 99% reabsorption is occurring. The areas of the kidney are defined. We have the outer area called as cortex, Sarika madam. The inner area called as medulla. पढ़ा कि नहीं? ये तो internal structure था ना, but outer area called as cortex, inner called as medulla, right? But can you see in medulla, the medulla is further divided, outer medulla and inner medulla. That means these are deeper parts of the medulla. So do we know and understand that this is going deep inside the medulla? If this is deep inside the medulla, what is this type of nephron? Juxtamedullary nephron. So what are the 2 types of nephrons we have? Cortical and juxtamedullary. Okay, so this is juxtamedullary nephron. Anything else to distinguish juxtamedullary? The presence of vasa recta. So if the capillaries are surrounding more around PCD, DCD, they are called as peritubular. And if they are more around the loop of Henle, it is called as vasa recta. Okay? Okay. Now, for counter current to achieve, right, the main thing is the arrangement of the loop of Henle and vasa recta. The main thing is the arrangement of loop of Henle and vasa recta. The diagram of NCRT, I am not a real fan of that. So, इसलिए we will take up this diagram. Right? It is shared in this PDF में है, तुम्हारे पास है diagram ये. Right? थोड़ा सा parts को identify कर लेते हैं हम लोग. This is the cortical area. Can you see this is the medullary area now? Bowman's capsule, PCD, descending limb, DCD, collecting. Fantastic. afferent, efferent. So can you see this is the other rector now? Around the loop of Henle. Okay. Now in the descending limb, is this the direction in which the filtrate is going?
Yes.
The downward direction? And in the ascending limb, is this opposite direction? That means the filtrate in both the limbs is in opposite direction. This is what is called as counter current. एंड ये सिर्फ डायग्राम में कन्वीनियंस के लिए दिखाने के लिए यहाँ पे ऐसा अलग अलग है, बट दे आर वेरी क्लोज टू ईच अदर.